Good afternoon. Thank you all for coming. I'm Barbara Snyder, president of Case Western Reserve University, and it is my pleasure to welcome you. Some of you are return customers to the Ford Lectures. One of the things I've discovered about being president of Case Western Reserve is there are many wonderful parts of my job. There are a few not so wonderful parts, but there are many wonderful parts. And I have to say that few things compare to the twice annual pleasure of spending some time with one of the finest citizens of our Case Western Reserve community and the special guests that he allows us to host. I am referring, of course, to Alan Ford. And I must be honest, I cannot limit myself to seeing him every 6 months. Fortunately, though, for me, he regularly attends meetings of our Board of Trustees. He joined the board in 1976 and served as its chair from 1987 until 1992. In recognition of his remarkable service and the impact he had on Case Western Reserve, he received the University Medal, which is the highest honor that our university can bestow, and he got that in 1994. Alan is now an emeritus trustee, and we are fortunate to benefit from his continuing engagement, his wisdom, and his passion for our university. One of the ways that Alan demonstrates his powerful belief in our potential is through his support of this lecture. He established the program to bring some of the world's most innovative thinkers to Case Western Reserve so that we could learn from them, and they, of course, would have the opportunity to see our outstanding faculty and students. In addition, the presence of these extraordinary individuals, people like Dean Kamen and Jeff Hawken, would in turn attract the broader community to our campus and to these lectures and give our visitors a chance to see some of our many strengths here. Alan, you know how grateful we are for this gift and how much we appreciate these opportunities for both inspiration and education, and I thank you publicly for that. Professor Jeff Dirk is going to provide the formal introduction of today's speaker, but I just want to mention one thing. Our guest arrived on Tuesday, and he has spent his time meeting with undergraduate students, graduate students, postdocs, fellows, faculty members across the campus, and staff members. And we are delighted that he has been so engaged in the time that he has been with us. Alan, I think that Michael West's itinerary fully maximized the opportunities that you envisioned when you created this program. So I think you can feel very confident that this has really fully realized your vision this time. So I say again, thank you very much. It is now my pleasure to introduce the chair of the Department of Biomedical Engineering, a research star in his own right, and the leader of a group of faculty members who literally change lives here on our campus every single day, Professor Dirk.
So thank you, President Snyder. One of the great things about being chairman of biomedical engineering is the opportunity to bring together the schools of engineering and medicine. Today's lecture is really another example of that opportunity. In specific, I'd like to acknowledge the partnership that exists for this lecture today between not only the Department of Biomedical Engineering, but our University Center for Stem Cell and Regenerative Medicine and its leadership, Stan Gerson and Deborah Grega. Without their support, this lectureship would not be as satisfying as it is today. And of course, I too would like to thank Alan Ford, the Ford Lecture Committee that's chaired by Horst von Rechem from our department, and of course our speaker, Dr. Michael West. A second great aspect of this opportunity of being the chairman of the department is the opportunity to engage with scientists like Michael, who's really provided us, I think, a lot of motivation with his title about the practical uses of immortality. Now, when I received the itinerary on Tuesday, I saw that in addition to the structured environment that we were going to have today for this lectureship, I'd had the opportunity myself to have some unstructured time with him and fellow friends like Deborah, Horst, Alan Ford, Stuart Rowan, and Joseph Wagner last night at dinner. And it was not until immediately before that dinner that I learned that I would be making this introduction. So my plan was to incorporate the things I learned in that dinner last night into some of my remarks today. So I want to share with all of you some of the things I learned last night. First, Gilligan, the skipper, and others made it off of Gilligan's Island. Second, it was Joseph Priestley and not Elvis Presley who discovered oxygen. Third, there is perhaps nothing that sends the chill down an academic administrator's spine like the potential link between immortality and tenure. Fourth, right, Barbara? Fourth, there are professors on this campus who change Wikipedia entries immediately before assignments are handed out to see if their gibberish appears on the students' responses. Do you want to stand up? And fifth, Michael spent time in northwestern Ohio searching for fossils as part of his early graduate career. Now, how are all of these different topics tied together? Well, we concluded first that the professor on Gilligan's Island had to be a biomedical engineer, because who else could make a radio out of coconuts? Second, as we study biology, it's not just the carbon, hydrogen, and oxygen that are building blocks, but also the DNA, the nucleic acids, and proteins, which really provide the clues to life and how life resides. Thirdly, as our investigators and investigations repeat the past and push the boundaries of the future, we are obligated as scientists to check our results and validate our facts. Wikipedia alone is not sufficient. And lastly, the motivation for Michael's research into aging and stem cells can be traced to picking up a fossil and asking the seemingly simple question, why is it that living organisms age? This question led Michael on a path where telomerase is utilized to immortalize human cells and to develop new oligonucleotide telomerase inhibition for cancer therapy through the founding of Geron. The company then supported the work of academics who isolated the first human embryonic stem cells and germ cells, among many other activities that we will hear more of today. Michael holds 64 patents, has published numerous papers in the biomedical sciences, and today he is the CEO of BioTime Incorporated and Embryome Sciences, companies focused on this next step, that is the research and therapeutic projects that are based on stem cell technology. If our own stem cell and regenerative medicine center is not proof enough of the importance of the stem cell field, I'd like to highlight the cover of Scientific American that came yesterday that says, "From your cells to stem cells, new cures may come from reprogramming any adult cell." So it's with great pleasure that I introduce today's speaker. Please join me in welcoming Dr. Michael West.
Thank you, everyone. It was amazing. All those things he said were true about last night's dinner. I can't believe you recounted all that. Listen, I want to thank everyone, the university, all of the faculty I met with, Mr. Ford, and all of you for such hospitality. It's been a long time since I've felt so much at home, and I very much appreciate the time I've spent here. Okay, so I'm going to talk to you about stem cells and immortality and all kinds of things. I won't touch on the fossil story here tonight, but so I'll start with my first love, which is the biology of aging. I'm a gerontologist. I think the biology of aging is very interesting. I'll try to explain to you the trail that led through my personal research, how it led to some of this telomere biology that we mentioned, how it led to the search to try to isolate human embryonic stem cells, and from a biotechnology perspective, which is the world I, for the most part, live in, how I see the future in the next decade or so. I'll try to give you my perspective on where I think some of the trends are and some of the opportunities, both for therapeutic product development and for basic research. So let's begin here. I bet some of you have seen this before. This is the coming age wave. It's one of those certainties of life, aging and death. And so here we see this trend. Look at here, 1900, less than 1 million people were over 80 years of age in the United States. We've seen this dramatic increase over time because of improved sanitation, antimicrobials, a lot of advances in medicine to this point. But look at this inflection point here, and look where we are in time. We're just now seeing it. This is the age wave. This is the coming tsunami of healthcare expenditures associated with an aging population. The major challenge for the economics of the United States, for clinicians, is the problem of aging. And I'm not here to talk about economics, but I would say that we would be foolish as a nation or as medical researchers not to recognize the challenge this is going to place for the United States and for many other developing countries. But how do we deal with it? Realistically, what can be done to develop novel technologies that would impact these healthcare costs or influence the health of people in the United States? So I'm going to take you back in time. Back to when I grew up in southwestern Michigan, back in the 1960s. And I'm going to play for you a video clip because it plays, I think it lays some groundwork to the points I'm going to try to make, and I'll see if any of you are old enough to recognize this. So here we go. Watch this.
Man, woman, birth, death, infinity.
Infinity, you should say. Do you recognize that, of course? Ben Casey. That's the opening scenes. And I was a young boy, and I was watching this program. And the next scene is, you know, the door is open, and Ben Casey's pushing a patient into the emergency room, and the music comes on. But as a young person, I looked at this, and I thought, what did that mean? What a strange intro to a medical thriller show. Can you imagine House opening up with something like this? And I looked at this and I thought, this looks like a hieroglyph, cartouche. You know, what is this? And of course, with time, I came to recognize it's a very profound principle of human biology, shown here. And I'm quoting here a German naturalist called Johannes Müller, who said, "Organic bodies are perishable," I guess we knew that, "while life maintains the appearance of immortality in the constant succession of similar individuals, the individuals themselves passing away, of course." And so here we have a daughter and her mother and her mother's mother's mother and her mother's mother's mother. And a scientist, a German naturalist named August Weismann, asked what I think maybe most interesting question you could ask. Why? Why is it that the human species continues to perpetuate itself for generation after generation? And he developed the first theory of heredity and corrected Darwin's incorrect concepts of how hereditary information is passed on. And this dichotomy between the germline, the reproductive lineage, and the soma has long fascinated humanity. And I'm a fan of this. And I know I didn't come here to lecture you on ancient Egyptian or Mediterranean worldviews, but I just wanted to point out the ancient world was quite fascinated with this. And here in Greek thought, you see the dichotomy described as Zoe, which was the immortal life of the gods, you know, the world the gods lived in, and bios, the individual life that passes away. And it's personified by deities in ancient Greek religion, like Demeter and Dionysus. You can see springing from the ground like we see outside right now. Every year, life reemerging in this immortal cycle. And the ancient world was quite fascinated by this and built their hopes, really, on their worldview, on this immortal renewal of life. August Weismann, we now move forward into the 1800s, as I said, asked the question of how is this Zoe, this immortal renewal of life, occur, and how does genetic information pass on from generation to generation? And he looked at simple multicellular animals like volvox and proposed that there were two types of cells here, probably reflecting an earlier evolutionary step where single-celled animals proliferated without limit and did not have to necessarily die. They could divide instead, and the individual cells go on to divide again and again. Although the individual single-celled organism could die, it didn't have to die. It could participate in this immortal cycle of life. But in the transition to simple single-cell or multicellular animals like Volvox, there was a branch point, a dichotomy, where some of the cells specialized into the soma, shown here, somatic cells, which served a reproductive function for its sister cells, perhaps protecting them from ultraviolet light damage or some other type of predation or some other, maybe, transportation, whatever it was. So these cells maintain the potential for immortality. These cells were programmed to die. And he proposed this was the evolutionary origin of aging and death, was the evolution of a soma. And Weissman, and here we see, again, the emergence of the young individuals while the shell cells, the somatic cells, would die. So here I'll show in some of these figures a simplified version of this, where these cells here would be the somatic cell lineages that are spun off from the germline, while the germline perpetuates itself in this potentially immortal merry-go-round carousel ride we call life. By the way, as a gerontologist, back when I entered the field, there was an inherent bias that the biology of aging had to be wear and tear. Free radical damage, other mechanisms of wear and tear. And what troubled me about all of these models of aging was, yes, but, you know what, you and I are made from cells from our parents, came from a lineage of cells that were in their parents and so on. A lineage of cells that have been proliferating since the dawn of life on Earth, uninterrupted. Continuing, never dying. Are the cells that made us have no dead ancestors. Unlike the soma, our somas, we have dead ancestors, but the germline leaves no dead ancestors. If it did, it would cease to exist. And so wear and tear, inasmuch as it occurs, had to be perpetually repaired for millennia. So why would we assume that the mechanisms that lead to age-related degenerative disease and aging itself are due to wear and tear, when life has evolved this amazing capacity for repair? So Weissman said in the 1800s, in a very prescient statement, death takes place in human beings because a worn-out tissue cannot forever renew itself, and because a capacity for increase by means of cell division, and forgive me, I should put in here, in the soma, is not everlasting, but finite. What was he saying? He was saying those cells that branch off into the somatic cell lineages in humans have a finite replicative capacity. And he even speculated on how that would work. He called this hereditary information the germplasm, and he said maybe in the germplasm there's some sort of a watch spring that's wound up every generation and then gives the somatic cell lineages this set proliferative lifespan. And I'm fascinated by this man. The degree of vision and rigor of his logic is quite extraordinary. He's often known as laying out the modern theory of heredity, but his insights into some of the biology of aging was really quite amazing for his time. Well, along came Alexis Carrel, and Alexis Carrel, shown here, was the first American to win the Nobel Prize for Medicine. I understand Dr. Millikan was the first to win it for physics. Dr. Carrel is at the Rockefeller Institute. No, this is not Al-Qaeda in black masks making anthrax or something. No, this is around 1910 or so at the Rockefeller Institute. This is how they did tissue culture in those days, in open rooms. He had them wear black, so he thought you could see dirt or lint better on black. It's a little bizarre. So he painted all the rooms black. This is how cell culture was done. He was one of the pioneers of cell culture. And Dr. Cornell published a series of papers saying Weissman was wrong. He said he had cultured cells from the chicken, from the embryonic chick, and that he could keep them proliferating for decades in his laboratory. And so however vertebrates aged, it was not because of an inherent clock of aging in somatic cells. It was some humoral or other influence of the organism that caused aging. And it caused Dr. Weissman's theory to be forgotten, put on the dusty bookshelves of history. He also, if you look, it's fascinating, he saw the vision of modern regenerative medicine. And so he wanted to, he believed it was, because he believed that the somatic cells of the human being had the potential to escape aging, maybe when cultured like he did in the chick cells or whatever, that we could make young heart muscle and young tissues of various kinds, and was a supporter or an advocate for what is now called regenerative medicine, but based on his own technologies. Well, then let's move forward in time. 1960s, Leonard Hayflick at the Wistar Institute was growing, this is WI38, it was a cell line he was growing to study cancer and to grow vaccines and various uses, and he was trying to grow them and expand them for various practical applications, and the cells kept doing this. And one day, as the stories I tell it, I hear it, I've heard different stories from Len. I tend to believe the story I'm going to tell you more. He was in a lunchroom at the Wistar Institute, discouraged over his problem with culturing cells, and he sat down at the lunch table, and one of his colleagues said, "Len, maybe your cells are just growing old." And a light went on. And he thought, you know what, maybe this isn't just an artifact, maybe this is an inherent property of cells. Len says he didn't know about Weissman's theory. It had long been forgotten. And he published what is now one of the most cited papers in the history of science, that human cells, normal human cells, not cancer cells, when cultured in vitro, have a finite lifespan, and we now call that the Hayflick limit, or cell senescence. And so that's when I entered the field. I was looking at these cells as a graduate student under a microscope. I remember the first day in the lab, my professor, Sam Goldstein, brought out some senescent cells, put them under a phase contrast microscope, said, "I've got to do some things, Mike. Why don't you play with these cells, look at them." And I looked down at these cells and thought, what in you is making this happen? I wanted to understand what caused this. What I soon learned was cells can escape senescence, human cells, with some modifications, with SV40T antigen and some other things. You can coax cells into immortal proliferation. By the way, what do I mean by immortality? This is Weissman's term. By immortality, he meant cells that don't have to stop proliferating. They can proliferate without limit. And if cells could be changed through probably mutations to suddenly reacquire what was probably something similar to the immortal characteristics of the germline itself, it can't be that complicated. Len Hayflick and others said, no. Cell senescence has associated with it hundreds of different changes. It's got to be too complex. We'll never understand it, and we'll never certainly be able to intervene in it. There was the differences of opinion. Then, in the late 1990s, I stumbled upon this paper. Howard Cook, a researcher in Edinburgh, had found a way to study the ends of chromosomes. And he was following this sequence near the end of the chromosomes called the telomere, and he noticed a heterogeneity, even though it was from one chromosome end, that he couldn't understand. And so he asked, when in human development does this heterogeneity in the length of the telomere on this one chromosome occur? And he published this figure. So in the germline, in sperm of men of different ages, this telomere was long and stable, but when he looked in blood cells, for instance, or other cells in people as a function of age in vivo, not in vitro, he saw a progressive shortening of the telomere in that cell, in that particular chromosome. And he published this paper. And it made me think. There was back in the late 1960s, there was a Russian scientist named Alexei Lovnikov, who was a theoretical biologist, who had just been to a lecture, somewhat like this, in Moscow, where it was being presented the work of Dr. Hayflick. Dr. Olovnikov, walking back through the streets of Moscow, going to the train station in Moscow, came up to the train, the doors opened, near the end of the train, and he said, aha, and he had it all figured out. And I've talked to Alexei. I have no idea how such aha things happen. He saw it all. As I could, clairvoyant vision. He said, and he wrote a theoretical paper in Russian, which hardly anyone read. And he said, what happens is every time the DNA replicates, I think I have a picture here, the DNA, the very ends of the DNA are like the end of that train. And he said, when the cells replicate, in Hayflick senescence, the DNA strands separate and are replicated. But like the people getting on at the door near the end of the train, these polymerizing enzymes cannot replicate the very end. And so every time somatic cells replicate, telomere is shortened from this defect. But there has to be something that maintains, rewinds the clock, like Weissman would say, this watch spring, in the germline and in cancer. That's something that makes this DNA at the end of the chromosomes. He called it the theory of marginotomy. Cellular gerontologists read the paper. We knew the theory. It had no experimental support whatsoever. I saw the paper, this result from Howard Cook, and I immediately said, that's Olovnikov. That's consistent with Olovnikov. This result I just showed you. I mean, that's Olovnikov's. The second thing was, I mentioned SV40T antigen. SV40T antigen will cause cells to go through in a longer lifespan and then rarely spontaneously immortalize. You take them to that extended lifespan, they start forming end-on-end fusions. The telomeres start joining together. And I looked at that data and I thought, that's Olovnikov. Could it be? Now, in 1990, some data came in from Cal Harley and Carol Greider that they cultured cells and saw progressive telomere shortening in cultured somatic cells. That's Olovnikov. So I was young, and I took a risk. And I started a company called Geron and said, this is the mechanism of cell aging and immortalization, and it has applications in aging and cancer, and we're going to go after the telomerase components to see, can we lift cells back to immortality? Can we make dermal fibroblasts proliferate without limit, or endothelial cells, and so on? And that was one of the primary goals of Geron. $40 million later, ouch, hard enzyme to find, because it's so expressed at so low of levels. We found it. First, the RNA component. So, by the way, if any of you are not familiar with telomerase, it's an RNA-dependent DNA polymerase. So here's the end of the DNA strand, and it uses an RNA template to polymerize the very end of the DNA de novo, off the ends of the chromosomes. And so we got the RNA component first cloned by guessing what the template sequence must look like, and PCR'd up a bunch of candidate sequences and eventually found one that we could demonstrate was requisite for telomerase activity. And we had the RNA component. When we looked, the RNA component had higher levels of expression in immortal cells, but marginally so. So we assumed that the immortal cells where the catalytic component or components, of course we didn't know how many, is somehow stabilizing the RNA, and it's there at a higher steady state level. It did not look like the RNA component was the regulator of mortality and immortality. So we had to find the catalytic component, and it was tough. And I won't take you through all the details on this, but Tom Check's lab had some students who were isolating telomerase from euploides. And fishing around in the databases one night, we found a small sequence from a human tonsil library that had homology, and the race was on. And for, I don't know, maybe 3 or 4 weeks, 24 hours a day, we were racing to try to be the first to isolate this catalytic component. Now, the fun part, and maybe even the scarier part, is to test the hypothesis. Well, that's a career-breaking event, isn't it? So you spend $40 million and you overexpress the catalytic component and nothing happens. Right? And that's what you kind of expect. And that's in the moment of honesty. I'll tell you a fun story. My wife will kill me for this. My wife was on the team that cloned telomerase, and we met there, and we were dating. And one night we were out for drinks in San Francisco, and she said, "So, Mike, is it going to work?" I said, "Of course it's going to work. Telomerase is going to work. When pigs fly." And, you know, she voiced that deep-seated feeling that, geez, of anxiety, you know. Science never works, right? I mean, it's not, right? I mean, you know. You're laughing because you know. There could have been 100 components to this enzyme. We just had one piece of it and all that. The fun thing was, I'll tell you another fun story. Len Hayflick was in one day at the company and being interviewed by a Canadian TV crew on doing a program on aging. And he wanted to show how you do cell culture. And so he hiked up his pant leg and cut off a little piece of skin off his leg to show how you take skin, and then handed it to me. He said, you want some cells? And I said, yes. So I want to measure the real Hayflick limit. So I was culturing Len's cells just when we had this expression vector made. These are not his cells, actually. I should have showed them. And so his cells were the first cells to see the catalytic component of telomerase and to show extended cell lifespan. It wasn't in the paper, but it was the first cells. Here's some other cells. So when cells normally senesce with the catalytic component of telomerase, the cells just kept going and going and going. And were not malignantly transformed, we had just overcome the Hayflick limit, and it worked. So the question I began thinking about is, what is the commercial value of this? How can we use these technologies to treat disease, to make money, and so on? And you know, I didn't know how to introduce the catalytic component of telomerase into aged tissues to potentially treat age-related disease. Maybe we could put them into a stem cell, a hematopoietic stem cell, and put back young blood cells into people and so on. And it occurred to me, I was always fascinated by the mouse embryonic stem cell. I wonder if we could get the human embryonic stem cell and telomerize it. We could make young cells of every kind. And I wanted to do that. But then I thought, you know what? If the human embryonic stem cell, which comes from the very nascent origins of human development, was prior, if the blastocyst embryo from which it could be derived potentially, if the cells in their cell mass were prior to the branch point of germline and soma, perhaps these cells would still have telomerase on and would be a naturally immortal cell, maybe the first cultured, normal, immortal human cell, and wouldn't need telomerase. That reinforced the idea even more. And so from Geron, we funded projects at Jamie Thompson's lab and John Gearhart's lab to get the embryonic stem cell and embryonic germ cells and so on. And then, of course, the cells were published in 1998. Now, the excitement about these cells were that they could My vision of it was that the cells could be used as a master cell bank of cells that would proliferate without limit and would not age, which is now, I think, amply demonstrated, and then be used as a master cell bank to manufacture many different products, many human cell types for therapy and other uses without limit. You would not have to go back and continually resource new master cell banks or working cell banks. And of course, you can make all human cell types, and I'd really emphasize embryonic progenitors. Primitive lineages like the neural crest and other cell types that I believe, my prediction, my bet, will someday be shown to have a potential to regenerate tissues not present in many cells in the adult human being. Now, so that was the beginnings of Geron, and we began to work up biotech manufacturing platforms. And the first one, and I'm calling it Regenerative Medicine 1.0, the idea was really carbon copying recombinant DNA. So imagine instead of scaling up CHO cells or bacteria or E. coli or something, we'd scale up embryonic stem cells in big stainless steel vats, turn a valve open and bring in FGF2 or something, or activin, and differentiate these cells into a desired cell type, but it won't be uniform. There'll be impurities. And so we'll weed these cells out using some affinity or other steps to make product. What I didn't appreciate, I think, back then, is the complexity of what we're talking about. And I've since gotten deeply into that complexity. These cells make, I believe, over 10,000 distinguishable cell types. I don't know where people came up with 200 cell types in the human body. It's not even close, and not even orders of magnitude. And we don't know the molecular markers or how to identify or distinguish these or what they're capable of doing when engrafted in the wrong place. So a few embryonic progenitors, a tooth bud progenitor or whatever, if put into the wrong tissue could form a tooth, and that'd be bad. And I'm showing a cyst here because many companies are seeing cysts showing up in their potential therapeutic formulations, and it's got a clinical hold on at least one company out there. And so we call that toxicity. It's sort of the parallel in drug development with stem cell research. There's cells that are not supposed to be there that can potentially cause inappropriate tissue types and so on. And this is a problem. I'll tell you, there's no debating it's a problem. It's had my old company Geron on clinical hold for an extended period of time. So how do we solve it? How do we determine identity, which the FDA really pushes for, of course, and purity, and then how do you scale a product while maintaining identity and purity? And the first generation technologies had this problem. I'm going to lay out for you an idea about how to change the paradigm. And it's an idea, and I'm happy to have you critique it. What I thought made sense is, given that there's so many cell types here, and because they have long telomere length, and because embryonic progenitors are generally highly proliferative, let's clone them. Let's grow up populations of cells from one cell. How are we going to do that? I'll tell you, if you just take some ESL colonies and try to clone out downstream derivatives, you pretty much won't get anything. So what we did is we differentiated them under an array of different conditions. Let's give them activin, let's give them FGF, let's give them this or that, different time points, and generate shotgun blasts over that tree. Let's enrich them in endoderm and ectoderm and so on. Take these candidate cultures and plate them out at clonal densities, but because we don't know how they're going to grow, let's grow them in, on laminin, on matrigel, in epithelial media, in endothelial media, and so on, and find cells that will expand clonally, scale them up. Here's an example of such a plate, and you can see where we circled some colonies that we'd picked. Here they are in the original colony and after 4 passages. And do microarray analysis. So we picked somewhere between 1,000 and 2,000 clones, scaled them up to roller bottles. If they scaled up and were stable and would freeze and thaw, we kept them as a line. Here are about 200 of them compared to ES cells on the very left. And I know you can't see the genes, but I wanted to show you, here are some markers that are overexpressed in these cell lines compared to the other cell lines. And I was wanted to, and they're clustered by relatedness here. The genes are here. And you'll notice that some of them we isolated over and over again. So remember, we're shaking the tree, whatever falls out. We're not trying to get any kind of a cell here. This is a shotgun cloning of the embryome, like Venter did with the genome. We don't know what we have. We let a computer reassemble it, so to speak. And now look at the, and I know this is just looking at a heat map, but you can see, okay, these are neurocrest, by the way. Some of them you got over and over and over again, but look at the complexity that you see in this heat map. And this is representative of the complexity that we saw. And so I'd isolated a bunch of cells, and as one critic told me, Mike, you got lots of cells. Yes, they're scalable and relatively pure, but they're cells in the search of a use. You don't know what you have. And that was right. But we had scalable, clonally purified product. And I felt we'll figure out what the uses are later. So then we began what we call, fate space mapping, where we expose these cells to different terminal differentiation conditions and did gene expression analysis. And I don't have time to go into all the details, but what I can tell you is, wow, what a gold mine. We found all kinds of novelty and things that excite us about these. And just to demonstrate an example, I'm going to show you, because this university, through Dr. Kaplan and many of you have worked so hard in mesenchymal stem cells, I'm going to talk about micromass culture and TGF-beta that typically makes chondrogenesis occur as one example of what these cells can do. So we screened these lines in chondrogenic conditions, meaning micromass culture with these factors. And out of in this case, about 100 of the lines, we saw these 6 as being chondrogenic, 4 of them, these 4 right here, being intensely chondrogenic, these 2 being mildly so, but under certain conditions, robustly chondrogenic as well. And I'm going to quickly show you some data on these. They're all highly distinguishable progenitors of human cartilage. Not 2 of them are the same. I think you maybe even see some of the heterogeneity in their morphologies. So here's one of them, 40-20.8 in a pellet culture stained for safran and O, and you can see the positivity here. Here's an MSC control and an adipocyte stem cell as a control. And they stained for collagen too. Both of them do. Now this line, 40-20.8, now the beauty of this is they're clonally purified, and what happens in this technique is that they turn on site-specific homeobox genes. So Hox genes and others are activated, even though these are very primitive. And so here, this line is very strongly LHX8 positive, and here in a mouse you can see LHX8 is very strong in the palate and in the mandible. And so here's the chondrogenic lines in your different time courses. I'm not showing you all the other lines because they're all negative. Here's LHX8. Normal human articular chondrocytes are actually negative. It's to the left of this line right here. Here's dental pulp stem cells from a molar that are positive. MSCs are negative. And here's 40-20.8 in an array of different conditions. You can see it's just pretty much constitutively positive for this marker. And then this is another line here. But you can see these other chondrogenic lines were negative as well. And then here's collagen 2 expression in chondrocytes, MSCs, this line, and several of the other lines. It's induced, only expressed in the chondrogenic conditions. Collagen 10, very strongly expressed in MSCs, not in chondrocytes. Very low levels in this mandibular mesenchyme, expressed in some of these other lines. So variability, consistent with the variability in these lines. LECT1, absent in MEL2, present in most of these others. Chondrogenic marker. There's another chondrogenic marker, very strong in chondrocytes. Pretty much absent in MSCs, at least in our hands, expressed at varying levels in these different lines. Indian hedgehog, a marker of hypertrophic chondrocytes, very strong in MSCs compared to these other lines. So I'm just giving you a sense of the variability here. CD74, which is a marker of mesenchymal stem cells, is also present in our hands in adipocyte stem cells, absent in our lines. So what this, I think, suggests, just in this area of cartilage, is the diversity of different kinds of cartilage that we could potentially generate from the clonal expansion of embryonic progenitors from pluripotent stem cells. And the ability to identify their site-specific homeobox genes and Hox genes and so on. You know, there's so much developmental biology literature, which is I'm extracting here, which tell us where in the case of neurocrest, where in the hindbrain these cells originate, based on this zip code signature that they're carrying with them. So what I desperately believe the field needs to do is to step back, both from the therapeutic side, I'm not suggesting a halt of clinical trials or anything, but think this through again about how we could redesign these technologies to make more purified and more highly identified products. The more highly identified has got a major problem. And we've been trying on our own as a little company to set forward in this project, and I've discussed with some of you here at the university, there really ought to be an international collaboration to map the Embryome. This is a front page of this website, but you click on one of these branches, we're taking it off into all the complexities of the thousands of cell types that originate out of ES cells. And we want to decorate this tree with all the molecular markers to help researchers identify precisely the different kinds of cells that they're making. Because I think the concept is the cartilage that makes the chondrocranium, or the cartilage that surrounds the trachea, these different types of cartilages, these different types of muscles, these different kinds of tendons and ligaments, and all the cells in our body, they're different based on site-specific homeobox genes. They're not all the same. And identifying all this is going to be quite important, we think, to the future of both therapeutics and basic research. Another major problem: how do we prevent transplant rejection, and how do we solve this ethical controversy about embryonic stem cells? As I've talked to some of the students, ethical controversies hampered this field. Boy, it's hurt us in biotech. You know, I was telling some of the students over lunch, I've had investors say, Mike, we don't know if this is the greatest thing to hit medicine or if it's going to be illegal, you know. And nothing was more controversial than human nuclear transfer, the idea of making patient-specific cells. My interest in it was, going back to this story about Hayflick, was can we take a cell from the human, aged human, back to a pluripotent state, but could we remember Weissman, rewind the clock of cellular aging and make young cells that we were born with, making young hematopoietic cells or other cells, especially where cell proliferative capacity is critical. And so we launched on this project after I left Geron at Advanced Cell Technology to do nuclear transfer. This is, you can see here, December of 2000. This is cow. I wished we'd taken videos of the human. Here are chromosomes being removed. That's the polar body. And we replaced them with cells that had aged in vitro, bovine cells that were senescent. Here's a whole fibroblast, skin fibroblast. And we refocus on the zona and plant the somatic cell within the zona, give it a little tap here to move the membranes in physical contact, and then electrofuse the cells and activate calcium flux, and the rest is the work of the cytoplasm of the egg cell. Well, we showed in 2000 that if we grew cells to senescence and used them as a source of cells, as I just showed you, in bovine, We started here with cells that were fetal in origin and regrew fetuses again and cultured the cells. They went even longer than in culture in terms of the Hayflick limit than the original cells, and their telomeres were reset even longer than they were originally. There was a lot of controversy about this. The group in Scotland said Dolly was born with prematurely old telomeres. They've since cloned many more sheep and have always seen a restoration of telomere length. I think that might have been an artifact. Dolly did not die from premature aging. She had a virus that is common, was common in that barn especially. Other animals that were not clones got the same virus, and it may be that it prematurely aged her blood cells or there was some other artifact. But cloning generally resets cell lifespan. So when IPS rolled around, we realized, as many others did, this is an embryo-free way of making therapeutic cloning work, patient-specific cells. The question then is, can IPS reset telomeres? Can it take cells back to pluripotency, but also back to immortality, so to speak? Now, the approach that we've been using is to use embryocarcinoma cells that overexpress Jamie Thompson's 4 factors as a source of reprogramming extract. But there are various, obviously, techniques to try and to use. Well, we recently published this paper showing that here's ES cells, here's telomere length, the normal germline telomere length in these 3 lines of about 15 to 20 kilobase pairs. But we started looking at widely circulated IPS cell lines, and their telomere lengths were really short. Cells senesce around 5 to 7 kilobase pairs, and so you can see these have aged rather dramatically compared to ES cells. And here are some other lines from Jamie Thompson's lab, passaged over time. I've summarized the data here. So whereas ES cells maintain germline telomere length, at least when cultured properly, these lines varied considerably in length, but none of them approached the length in our hands that we saw in normal human ES cells. When we look at telomerase activity, this is the TRAP assay, PCR assay, you see this ladder of telomere lengths. And ES cells showed slightly elevated telomerase activity compared to IPS cells. Here I'm quantifying the activity here. Is this the reason? I don't know. I simply don't know. We summarized in this paper what we knew about it. So what we decided, though, was critical because telomeres are very heterogeneous. Let's take H9, human embryonic stem cells, clone out, like I just showed you, these clonal embryonic progenitors. Take these clonal lines derived from the ES cells, and they were all mortal telomerase negative cells, and use them as a substrate for IPS, because now we know what our target is. We know precisely what we want to try to hit. And so we used retroviral-mediated IPS transcription reprogramming. And here are some of the clones we looked at, and we saw these. You can see this line here. Here's the progenitor lines, telomere length, maybe a slight increase at passage 3, but drifting down, certainly not increasing back to an average TRF length of 15 or 20 kb. But we found one clone. Look at this. Isn't this pretty? August Weissman would be proud. Here is ES cells, germline telomere lengths. Here's an aged derivative of it, and in the presence of these transcription factors, a return back to germline telomere length. Now, I don't know about you guys, but I think that's magic. I just think that that's really amazing. So, and again, like cloning, the cells, they stabilized now, and they've stabilized in telomere length above what they were in the original H9 cells, sort of like in cloning. And of course, these cells are capable of pluripotency and making all 3 germ layers. So this may be elementary for many of you, but the concept here and the vision here for regenerative medicine, gerontology is, we might have within our hands finally a means to take a cell from a patient back to pluripotency, but also resetting the clock of aging, in a sense taking the cells back to immortality, and then making young cells of various kinds specific for the patient. And This is a technology. I asked Len Hayflick, I showed him some of this data. Len, did you ever imagine that medicine would have within its hands the potential to take cells from an aged human back in time and potentially unlock some of these molecular mechanisms of immortality that allows the perpetuation of the human species, but to use it for the perpetuation of the individual human being? And as you can imagine, Len agreed. Never in his wildest dreams. And what I think is that there's a great opportunity here with this age wave coming to apply some of these technologies. Maybe we have just enough time, maybe about 10 years, to apply these to some concrete ways that could save our nation literally trillions of dollars for the treatment of chronic, debilitating diseases of aging. So here are the ES cells, H9. I thought you'd enjoy a little time-lapse video. Oops, a little time-lapse video. If I can find my cursor. There you go. Here are H9, a colony. You notice the prominent nucleoli and the scant cytoplasm proliferating on matrigel, so we can see them, so they're not, those of you that are experts in ES cell culture, you could say, Mike, I've seen better colonies. Yeah, I know, this is on matrigel. So there's H9. Here is EN13, the Splanchnopleuric mesoderm that we had aged and used as the substrate that was H9 derived. They look like they're busy bodies, crawling around in the dish. And then I'll move on. Here are re-H9, as we called them. Here are cells taken back to pluripotency, back to immortality, resetting cell lifespan. This is the line that we saw this restoration of telomere length in. Kind of pretty, I think. Now, I talked about, some of you asked, Mike, what is the practical uses of immortality? What I mean is the ability to tap into the germline, reset proliferative lifespan is, I think, obviously important in the treatment of many age-related diseases. But some of the practical uses, and I'm hopeful that in the coming decades, maybe 100 years from now, people lecturing in this university, in this platform, would be saying, you know, the ability to have an immortal substrate, the ability to reprogram cells or to use human embryonic stem cells that can proliferate without limit, allows us now to go in with targeted genetic modifications like we do in the mouse and do homologous recombination, remove a gene, put in a gene, expand the resulting engineered cells in a new population of cells, do it again, and now we're doing evolution. The immortal germline, modifying over time, but under the hand of man. And what we can potentially do here, I think all of you would recognize, is really only limited by the human imagination. We can make all human cell types at any point in development genetically modified in any way the human mind can imagine. And so I think that would be a practical use of immortality and a potential benefit to our population. So there's a lot of opportunities in pluripotency. There's a challenge of the complexity. We're dealing with very complex anatomy and development and genetics. We have to get a handle on that. But the potential applications in age-related degenerative diseases are the demographic trend of our time. It's going to be the story of the decade. And so I think whenever you see opportunity like that, I think it's imperative for us to seriously consider it. From a business standpoint, when you have a confluence of supply and demand, a technology that could potentially benefit such large and growing markets, there's great opportunity. And so in numerous ways, I think we should seriously consider how to advance these technologies. I was in New York and I saw this, and I'm a gerontologist, and I could have swore that said I'm aging, but if you blink, it says imagine. And that's really what I wanted to leave you with today is my view into the future and imagine how we might, all of us, be able to make the world a better place. And I want to thank all the people who helped with a lot of this work over the years. Jose Sabelli did nuclear transfer, and telomeres were done by, among many other people, Jerry Shea and Woody Wright, Cal Harley, Aman Vaziri, who's on our paper now, Jamie Thompson, of course, with embryonic stem cells, and some of my colleagues here, and my wife, Karen Chapman, with a lot of the embryonic progenitor work. And thank you very much. Did we want to do any question and answers, or do we have any questions? Not really? Yeah.
Michael, if the developmental program continues throughout our entire lifespan,
Yeah.
It means that those early embryonic progenitors aren't existing in the older individuals, so that concept of backing up becomes precluded by that logic. I wonder whether the whole area of tissue engineering, the whole area of transplantation, surgical transplantation, is going to have to be emphasized in order to do these corrections that you're thinking.
Right. Almost certainly. And the examples I give, because I know many of you here have been thinking about orthopedic applications. An example would clearly, I think, be in the case of cartilage, combining of cells like this with matrices. We did some work years ago at my previous company, Advanced Cell, on making retinal pigment epithelium. In aging, the retinal pigment epithelia senesces and becomes lost, and these cells serve multiple functions, but one critical one is the angiogenesis inhibition, keeping the choroid cells, the vascular network, out of the eye. And when they're lost, that's lost, and you can get a sudden invasion of vasculature and the wet form of macular degeneration and so on. And so we saw that you could easily identify these RPE cells because they pigment. They're pigmented patches or freckles on the dish. And began some animal preclinical work on those cells, and saw in the rat model, the RCS rat model, promise in preventing retinal degeneration. But when the cells were applied to a membrane, instead of just injected into the subretinal space, dramatic improvement in their ability to retard neovascularization. And you can think of numerous examples where these cells, although these cells, I didn't show you, I guess, but they have the ability to organize themselves when you have proper mixtures of cells into hair follicles and other intestine and other kinds of tissues. Nevertheless, in many practical applications of immortality, so to speak, combining the cells with tissue engineering and existing technology is going to be the smart and judicious approach, don't you think? Long-winded answer to your question. Yeah. Do you have any other questions? Oh, yeah, sure.
Doctor, you talk a lot early on in your presentation about the trends, about how there was a discovery at some point of an earlier form of multicellular organism, which sort of marks the first time you encounter an organism that has the ability to age. Since from an evolution standpoint, success is measured by how likely a species is to reproduce, in all your experience, have you ever encountered maybe the what advantage the process of aging lent to reproductive success that would make it so prevalent in so many organisms?
Yeah. There have been people who have looked at that. In the University of Sussex, I'm trying to remember, Maynard Smith. Michael Rose in California has written some books on the evolution of aging. There are people who looked about it. I think the consensus, though, is we don't know. There's thoughts that in insular populations, in some island somewhere, and there's some circumstance, the programmed aging of one organism, like the salmon, the sudden senescence of the salmon after spawning, may have provided a reproductive advantage to its offspring, and then that spread throughout the population through some chance, isthmus forming, and then it spread throughout the world. You can build models like this, but without knowing precisely every aspect of the evolutionary history of life on Earth, which we are not in possession of, it's hard to speculate. There's one thing we can say that many people in the field buy into, and that is antagonistic pleiotropy. And that's the idea that early in vertebrate development, maybe when we were a small mammal, we don't quite know, there was an advantage to living longer. And there's all these theories about why humans live as long as they do. It's because of our increased cephalization and so on, neoteny and all these theories. And the idea is, okay, so there was a selective pressure for an extension of the human lifespan, and, well, we'd get cancer. And so now we have one pressure pushing for an extended human lifespan, and then another selective pressure now to repress malignancy. Mice oftentimes get malignancies within 2 years. And so the thought was, well, okay, let's shut off telomerase very tight, set telomere lengths. I say this, teleologically. Obviously, nature didn't think this through. And but increased telomere length and then a tight repression of telomerase might have helped, and many other changes, shutting down, by the way, many of these regenerative capacities of the early embryo, so that we can't no longer regenerate limbs and things. All these things might have made malignancy less likely, and but the consequence is that in modern times, when we have the ability to get better nutrition and live longer, our tissues start undergoing cells senescence. And by the way, I know it's hard to cover all these things, but an important thing to think about, when cells get shortened telomeres and they senesce, they don't just stop proliferating. There's a change in gene expression. The cells move into an inflammatory state. They're saying genotoxic damage, something's very bad wrong, and many of these are downstream events of senescence that explain many of the aspects of age-related pathology. But so, yeah, less cancer, we live longer, but it kills us. Antagonistic pleiotropy, it's called. Yeah. Well, thank you. Thank you. Thank you.
Stay right here. We have a gift for
Thanks.
you.
Okay.
We have a gift for Professor West. We always like our guests to go away with something to remember Case Western Reserve and their experience here. So this was done by a local artist, Erica Weiss, and she does these for us, and we try to select them so that they will be something special, somehow connected to your work visually, if not scientifically. So we hope you will enjoy it. I invite all of you here to please join us outside for a reception and an opportunity to speak individually with Dr. West. Thank you for coming.
Thank you.
Thank you again.